Good morning. My name is Sebastiano. I'm a first-year postdoctoral fellow at the Eric and Wendy Schmidt Center, and it's my pleasure to introduce our next speaker, Jean-Philippe Vert. Jean-Philippe is the Chief R&D Officer at Owkin, an AI biotech company using machine learning to discover and develop treatments for unmet medical needs. He's also the co-founder and executive chairman of Bioptimus and professor at the École des Mines in Paris. His research focuses on the theory and practice of statistical machine learning and its applications to computational biology and medicine. JP has earned his PhD in mathematics and has held roles at Google Brain as well as academic positions at the École des Mines and Institut Curie. His contribution to the field has led Jean-Philippe to several awards, including a major science prize from the Institut de France. JP, we're very excited to have you here, and we're very excited for your talk.
Well, thank you for the intro and the invitation to be here. It's a real pleasure, and it's always so. I mean, these are my best workshops. Where not too big with real AI, real biologists meeting together. This is where things happen. So I'm gonna give a presentation which comes, compared to the previous one, a bit more from the AI viewpoint, because this is me. I cannot pretend I'm a biologist, and I'm gonna discuss, a topic which I think is really mainstream now, which is the National Foundation models and how the latest AI technologies could contribute to different ways or new ways to understand, simulate biology. First, let me mention, because so I have academic position, but I'm now in the industry. Just for all of you, maybe not all of you are aware of what all of this means in industry. Lots of biology, especially in such an institute, is geared toward developing new treatments, better treating patients. And so I must say that already today, and it's been the case for the last few years, AI is used and deeply used in many aspects of drug discovery, development, diagnostics, etc. Forget about the details of that slide. This is just a slide taken from Owkin, but it shows that Owkin is using AI in order to solve deep problems in drug development, and really machine learning, computational biology is used from the beginning. So developing a drug means understanding biology. This is mostly what is done in academic labs, right? To understand cancer, for example, identifying processes, targets. So this involves also computational approaches. Then the second step, often called drug discovery in these approaches. Once you have a target, design a good drug, it could be a small molecule, could be antibody, this kind of thing. So obviously today Gen AI is heavily used already to design candidate drugs. But then it's not the end of the game. Of course, once you have a candidate drug, you optimize it, and there is all this translational work, which is where should I use my drug, to which patients, which are the likely responders, etc. And here again, you can think of it as a massive, collaborative filtering problem where you have patients, you have candidate drugs or candidate targets, and you want to find a good match. So just for you to know, and each of these topics could involve, a full day of seminar, but AI is already used and with some success. However, it's fair to say that in spite of all the smart people you doing that, all the great data we have, the statistics are not very, very good. And typically, after years of development of a new treatment, finding the best science, the best antibody, the best small molecule optimized, you test it in clinical trial, meaning you test it on humans to test if you will save lives. And we're still today at the stage where 9 out of 10 drugs that are tested in clinical trials don't make it to the market, either because they are toxic or because they have no effect, they don't treat the patients, or other, more business reasons. But so something is missing, and the whole. By the way, this is very high level, but this means that in spite of the great science that we are so far away from other fields, like in physics, where we have equations, then we can fly rockets to the moon almost. Here in biology, we are still probably missing deep understanding and capacity, computational simulations, prediction models that translate from research to real patients. Okay, so I would summarize my presentation by okay, this is a fact, we have lots of knowledge, lots of experience, but no real computational simulation that would allow us to just say, well, developing a drug is easy, you just find a target, and then your computer would simulate if the target works on a patient, and it would. Today, it does not. Okay, in that field, this AI is a hope. There are many ways to introduce it, but all of you have heard and probably maybe have developed or are working on things like AlphaFold. This is a big game changer. I just mentioned it because it's popular, and also just to illustrate again, if it was not obvious to all of you, that something has happened in the field. This is a specific problem in biology here. We're talking of predicting the 3D structure of a protein from its amino acid sequence. And that's a field where, again, there has been lots of smart people developing smart ideas over the years, but where the performance of the prediction was plateauing, has been plateauing for more or less a couple of decades. And then came a different approach, AI-based approach, big neural networks, which borrows idea, of course, from what was done before, but pushes it to a new scale and with a few new technologies that really changed the field. Suddenly the performance is better and turns out to be useful. Something important is that when I talk of new approaches of AI, and this is what I will now discuss in my talk, there is something that is interesting, which is that a lot of the recent progress in AI in biology, like AlphaFold and beyond, is based on approaches that are slightly different from what machine learning was 10 years ago. And it's really a lot of emphasis on purely data-driven AI, meaning if you have lots of data, then it's possible, we discovered ways to train neural networks or machine learning models in an unsupervised way, so I can call that self-supervised or unsupervised, but meaning with no human annotation, with no specific, objective function in mind to solve a problem, just looking at data and managing to learn from data a good representation, a good simulation of the data, so that then it can be fine-tuned or used to solve a variety of tasks, right? So this thing is my definition of a foundation model. Foundation model is really something, so neural network typically that is typically very complicated. So it's not like a few simple equations. It's really billions of parameters which are optimized, but trained just to look at data, a bit like biologists have done, and scientists have done forever. Look at data, then eventually do hypotheses, do experiments, look at more data, and aggregate this knowledge into your brain. Today, we try to do the same, but the aggregation of knowledge is done in a computational system. Okay? So, of course, this idea maybe 5 years ago would have been, it's a cool idea, but it will never work. I think all of us here, probably every day you use some LLM, some Gemini, GPT, etc. And suddenly, something that sounded crazy 5 years ago is not crazy anymore. An LLM is just a foundation model of language. So GPT, Gemini, all these things, they are just systems which have read texts taken from the web with no specific things in mind, but have tried to learn to speak just looking at raw text. And what we realize today is that they speak pretty well. It's not perfect, I won't enter into the question of whether this is intelligence or not, but at least it's a very powerful system that just from raw data has learned to answer questions, has learned to generate text, etc. So the question is, could we do the same in biology? And this is, it could be a bit provocative to say, well, maybe we don't need so much humans there. Maybe if you just collect lots of data and find a way to train this equivalent of LLM, but from raw data, what could they learn, and could they build a system, computational system, that then can be queried, can be used, to address many questions. The previous talk, I think, gave a few examples of how this can be done if you want to identify structures in a cell, but maybe we could pre-train a system that then could be queried up to, can you design me a good drug for these patients? We don't know. Okay, so we are not there yet at all. It's very hard to know whether we will get there, but I think there's lots of progress being made. Something more complicated in biology than in language is that, language language, at least there is one modality which is text, which is the modality used to train LLMs. In biology, as soon as you say, let's train a foundation model of biology, first question is, okay, what is biology? What are we talking about? Are we talking about molecules, cells, tissues, organisms, ecosystems, etc.? So there's lots of levels. There's lots of modalities. It's a mix of images, genomics, etc. So it's a broad field where I have my me and my teams we explore it, but it's like there is an endless combination of things we could try, and I think this is going to be a very exciting topic in the years to come. It already started. I mentioned AlphaFold, but I think these days, if you follow bioRxiv, etc., every week you have new foundation models. I would say that as of today, most of the foundation models and most of the publications that come out tend to be on specific modalities, specific levels. So you have, I will go through a few examples, but we hear a lot about foundation models for small molecules, for proteins, for single cells, for tissues, etc. By the way, the field is really growing fast. There are hundreds of citations, so I show a few examples, but please pardon me if you're not with work there. They're just, it's just a mini sample. But just to say that these high-impact publications are coming, and they really have, each of them is really bringing lots of novelties. And when you look at the results, at least for specific tasks, we see that this approach seems to be working in, maybe not yet in all dimensions, but I will give a few examples of where it really brings a new performance improvement. Okay, so foundation models of biology are coming. Let me deep dive to a few of them before then coming back to how we build a more unified view of biology. So I would just mention, and again, I take risks here, especially in front of this audience, because I will cite some work which are not mine at all, which I found interesting, a bit outdated, by the way, just to show that it's already there. So proteins maybe has been the first, there was AlphaFold and then lots of work training LLMs on proteins. One good reason being that we know how to train large language models on human language. If you replace a text by a DNA sequence or an amino acid sequence, you more or less can use exactly the same architecture, the same transformer. Click the button, it learns to speak not the language of humans, but the language of biology. So these work I mentioned here are a bit old already in that field, but they demonstrated that in principle, just doing that gave new capacities like generating proteins. You can query proteins, you can predict the impact of mutation, etc., to some level. Okay, so this is already coming. Of course, there is this super exciting field of single-cell genomics, and I'm sorry again, I cite only one paper out of hundreds. I know the broad and many people here are deeply involved in that field, but the fact that we have access now to the capacity to generate large volumes of single-cell transcriptomics, for example, we're talking of tens or hundreds of millions of single cells, suggests that we can learn the biology of single cells, or at least try to build a foundation model of single cells that could allow us to have a virtual single cell, once trained, maybe allowing it to predict the evolution of cells, time evolutions, or the response to perturbation, etc., etc. So there's lots of work, and there are many people in this room much better than me in that field. But this is really, a great example of how the current AI technology can really match the current biotechnology and biology topics. New technologies, lots of data, and a great object to study, in this case, single cell. Now let me spend a little bit more time on what's beyond cells, because understanding one cell is good, but if you work in cancer, for example, everybody knows that it's the interaction of cells, complex interactions, tumor microenvironment, etc., which matter probably more than understanding what one cell does in a petri dish. So how do you go from one cell to many cells, to interactions between cells? Here there are many technologies, but when you think of how to get large volumes of data, it's often, there is single cell genomics for single cell, but for tissues, something that is quite common and for which is very easy to get lots of data are images, in particular these types of images in histopathology, because this is what's used in many clinics in the world, right? As soon as you have a suspicion of cancer, you do a biopsy, you look at under the microscope. And if you have a scanner, then you can make it digital and suddenly collect millions of images. So, of course, the depth of the information maybe is a bit shallow because it's only an image. But remember, these types of images are what is used today by most clinicians to do diagnostics and often to decide the treatment, right? So there is information there. So the question is, if you have this data, well, could you build a foundation model on that would mimic, in a sense, what pathologists are learning, right? You look at lots of images to understand the diversity, the patterns that you can have, and maybe then, from an image, do a lot of prediction tasks. Maybe I just skip that. So just to anchor again this comment in the real world, this is one example out of many of systems that already exist and already use foundation models to help patients. That's one system which is actually that was developed by Owkin, so which is used in the clinics for one task. In this case, it's in colorectal cancer, from the image to predict the status of microsatellite instability, which is a good predictor of response to immunotherapy. So this is something that is important for clinics. There is a genetic test that is the standard to decide of the MSI status, but adding to the genetic test a pre-screening phase by images can be a way to accelerate, right, the filtering out patients who are likely to be positive or negative. So that's the kind of thing that that finds its way in the clinical protocol. And this thing is just one machine learning application where you have an image as input, a prediction to be made, MSI positive or negative. And that's typically the kind of things which already today benefit a lot from these foundation models. I will show some results. But in the same way as it's a machine learning problem, but boosted by the recent approach, the modern AI approach, to say if you can pre-train a model on millions of histopathology slides, then you can also improve the performance of downstream tasks like this task, which is already in the clinics. Okay, so how do you train foundation models? If you're, I know this is a mixed audience. In LLMs, like in protein sequence, natural language, most people know that it's just trained by predicting the next word from the context. So you see lots of text and you train a statistical model to say, well, if you have seen this thing, what is the next word? And if you know how to predict that, you get an LLM. Of course, for images, it's a bit more different because there is no notion of sequence. But just for your information, the field has tried many ideas, has converged to a few that seem to be working. So FYI, the kind of thing that seems to be working today is to mix. So it's, again, it's self-supervised, meaning here I'm just talking of, suppose you give me millions of images. What could be an objective function for a neural network when it sees the images to optimize the way to do something, okay? And the things that seem to make sense, that seem to work at least, is, as of today, mostly a mix of two main ideas. One is called contrastive learning. So it's the idea to say, if you have an image of a tumor, then by yourself or through different measurements, you can take a picture of the same patient or the same tumor, but changing the color, changing the orientation, changing a bit the geometry, and just constrain, so teach, let the neural network learn that it's the same tumor, right? So in a sense, learning the biology and not the artifacts of the thing. This is one key idea. The second key idea, which is quite similar to what's done in autoregressive LLMs, except that it's not a sequence, is just what's called generative learning. So you take an image, you hide part of the image, and you train the neural network to reconstruct the hidden part. This can be done without human intervention because it's purely computer that takes an image, removes part of it, and trains the model to predict what it has removed. So this is really this notion of self-supervision where no human intervention, the computer can do it by itself and do it at scale. Okay? So when you do that, and then you can train a foundation model of pathology. And once you have done that, what it is at the end, there are many ways to query it, to use it, but one of them is that the model has learned to represent images, so it can read an image and map an image to a representation in numbers, right? Fixed vector representation, often roughly about 1,000 numbers. And then from this representation, you can fine-tune it to solve tasks. So for example, I mentioned the MSI prediction is one such task, meaning if you have a smaller set of images with annotation, you can fine-tune your model so that it can predict MSI. Here I show one work, so I think it was one of the early work, one of the first foundation models for pathology, which was done at Owkin in 23. It's called Owkin. And so we developed here, it was a foundation model for pathology trained on TCGA data, basically public data, so not big, but not super big, I would say. But already when we tested it after fine-tuning on a number of tasks, and here I won't go to details, but it's a mix of, from an image, you can predict a variety of genomic alterations, like BRCA mutation in breast cancer, for example, or you can fine-tune it to predict survival, so for prognosis, or predict molecular subtypes. Systematically, we got some big improvement compared to what was done before, which is more standard machine learning. And by the way, just to go beyond internal validation, we also tested it on a Kaggle challenge. So I really like, challenges like Caroline mentioned. This is how the field can, progress by step, because after a challenge, everyone observes what works, what doesn't work, and we can follow up on the best ideas. And so at the time, there was one challenge on ovarian cancer subtype classification organized by UBC that we won, actually, with quite a margin. And basically, the main difference between our approach and the others were the use of this foundation model. So much less impact than AlphaFold, but in a sense, similar pattern to say, well, if suddenly you decide to take a different approach, build foundation model and check them, they just improve the field, right? So, and it's systematic across a lot of applications. Now, once we were there, of course, there was this exciting question, say, well, we have trained a model on TCGA, but all the LLM field tells us that if you grow your model, if you have more data, then maybe the performance will improve again, which is something we don't understand perfectly why, but it's intuitively the case that the foundation models benefit from volumes of data. Okay, so that's what we did, and this is one of the reasons why, by the way, we created this new company, Bioptimus, which is solely focused on developing foundation models for biology. It requires some, capital. It's expensive. Therefore, the idea to have a VC, some capital. But so we released last year some models, but more recently last month a model which we call Optimus 1. And just to give you an idea of the scales where we are today, this is a model that instead of 5,000 images from TCGA is trained on more than 1 million. And importantly, coming from many patients, right? So to get patients, we did partnerships with a variety of labs. We worked with more than 4,000 clinical practices. So we are I'm sure we can go beyond, but already we moved from thousands to millions. If you're in the machine learning field, important is that here we're talking each image is very large. It's a gigapixel image. So in fact, to train the model, we cut each image into thousands of smaller patches. So the number of small images that the computer sees to train the model is in the billions. Okay. So we did that. It's available on Hugging Face if you want to try it. And does it work? Well, we played, we tried. So by the way, I just mentioned our work, but that's a field that is booming as well. There are lots of other very nice models that have been proposed over the years. But just this is some of the results. So I won't go to, there's a link to where the results are detailed. But this is a summary of 18 tasks where you say, I have trained a model and I use it to predict genomic alteration, prognosis, classification, etc. Then you can measure the performance, if it works well or not. And here, these slides aggregate the performance, right, AUC. I think there are a couple of things to be said here. So one is every column is a different model. The one I presented earlier, that one Kaggle last year, which is quite important, it's not even here, is a predecessor of Owkin V2. It was Owkin V1, right, which is much below there. So first comment is that between end of 23 and 25, we moved from, let's say, 80-ish to 85-ish by just changing the foundation model, right? No other innovation. You just change one component of your system, all the rest is the same. And you see that we're in a field where the progression in performance on tasks that matter can be in the range of 5–10% easily just by changing the fundamental. So I think it's not the end. I don't know how far it can go if it's a question of more data, bigger models, other modalities, etc. But as we speak, and this is what I was saying, where I said every week there is a new model, a new publication, we're in a very active field where we see progress, which hopefully impact the patient. This maybe I can skip. Because now I'd like to, to take the last 5 minutes to discuss what's next. So I insisted on foundation models for images. I mentioned many people are doing things. Now the real question, the real exciting question that I'm sure many people share is, okay, suppose we have a virtual cell, we have a virtual tissue, we have a virtual protein. How do they fit together? And remember that on the application side, the real question we care are, how can you design a drug for a patient? So it has to be a good protein that changes a cell in the correct way, in such a way that in the tissue, in the tumor microenvironment, it ends up with benefits at the clinics, right? So these things are connected. They have to be connected. This is exciting and hard. I can quickly share some of the avenues we've taken already. So, to go across modalities, there are questions of how do you get data that connect scales and modalities. Something that, in terms of existing technologies that we started working on, is spatial transcriptomics, which is a good bridge between single-cell genomics, single-cell transcriptomics, and tissue organization. And in particular, one thing that you could decide as a task is, suppose you observe an image, could you design a system that allows just from the image to reconstruct the genomics, so that eventually, the idea to go that way is that because the pathology is cheap and in large volumes, if we had a way to, from the image, reconstruct much more than the image, but maybe some gene expression, etc., then suddenly we could map millions of images to that genetic level that would allow us to investigate a correlation between organization of gene expression and response, for example. So, we worked on that, and it turns out, I don't know if Faisal is in the room, maybe, but there's been a great paper at NeurIPS last year, which is a benchmark, super useful, not a challenge, similar to a challenge, that proposes one such task, which is collecting images, and among other tasks, from an image, can you predict gene expression? We did test our models. They work very well. Again, these are many models. Ficon is here. You see, this was top-notch 2 years ago. Now it's quite behind, so the field is moving fast. I think there are two comments here. One is that, again, in 2 years, we go from 36% to 42% performance. I skip the details. The performance here is a correlation coefficient between the predicted and the real expression, average over many cancers. But so there is big progress. Still, I must say that the raw performance, 42, is very far from 1. That would be the optimal. So there is a long way to go. Probably it will keep growing. Probably there is a limit, and so probably we cannot predict all gene expression just from H&E, just because there is not enough information. But I think there is still room for improvement there. Okay, so it's a good first step, and here you really see the benefits just of training better foundation model, because again, all the columns is the same algorithm. It's just you change one component, which is the foundation model that you use. How can we go further? So a couple of ideas. One is that we need more data. I think any initiative to systematically generate and share data is welcome. One of them is a very cool data set being generated sponsored by Owkin, but with several hospitals in Europe and in the US to systematically generate an atlas of cancer, in particular using spatial transcriptomics technologies, together with single cell, bulk, whole exome sequencing, images, etc. So that's the kind of data generation that I think will really support the field of AI and biology. And again, foundation models are really ready to ingest all of this data, take this data, they will just improve the models and move us maybe towards more multi-scale, multimodal foundation models. And I will finish saying, so, we are working hard to release the next generation of foundation models. Unfortunately, I cannot go to too many details until the product is launched, etc. But the real thing we're working on, and I'm sure that I would love to have discussions and see other ideas, is how do you, integrate these modalities, images, genomics, patient records, proteins, given that we have a beautiful system in biology, which is this multi-scale approach. We know that what you see in full body is a consequence of molecular interactions, but the path from one to them is well understood conceptually, but it's a bit hard to model. So, one idea, for example, is that because each foundation model, you can see it as transforming building blocks into one object, like a foundation model for proteins is how amino acids together from a protein. Then if you look at single cell foundation models, it tells you how a set of genes and gene expression defines the phenotype of a cell. When I mentioned the tissues, it's a way to say, well, if you know what each cell is, then when you put them together, you have a representation of the tissue. So computationally, mathematically, it seems to make sense to have some hierarchical structure in some neural networks. So this brings questions in AI about what is the good architecture of the model, but also what is a good objective function, what is a good tokenizer to transform the raw data into building blocks, etc. So lots of cool ideas, and I'm talking especially, I understand there are many postdocs here. I think there is enough research for all of you if you want to go in that field. Thank you. I'll stop here, and I will thank the teams at Owkin and Bioptimus who did most of this work. Thank you.
We have time for one question. I'll go here.
Thank you for the great presentation, Jean-Philippe. You mentioned there's new models coming out every week, and perhaps the consensus on what the best model for a given workflow might also change at a similar frequency. How do you ensure researchers are constantly equipped with the best model? And what are the biggest bottlenecks to quicker and widespread adoption of these models, making sure that they get in the hands of the researchers that can apply them for their workflows?
Yeah, great question. So, what I describe is, I think, just the fact that this is a fast-moving field. So, there are the people who develop these models, and there are the people who use them. So when you ask the question, how can a user be sure to use the best model? I have no answer. I think you should just take the ones that are available, that exist today, knowing that, I don't know if it's a good or a bad news, but probably in 6 months there will be a better one. So, but in research, you just cannot say, well, I will wait 6 months to see what's the best in 6 months because it's just moving too fast. So, maybe a good answer is, I think these benchmarks, these challenges are really helpful. And I've been doing research and science for quite a few years as well. What's complicated is when you just look at publications, individual, each of them saying, well, look, my model is the best. We've seen, this was the case for AlphaFold, with the competition CASP. Even though everyone can claim their model is the best, the day you have a competition like CASP that says, look, there are many ideas, but one of them is just so much better than the other ones, the year after, everyone is using it. Is it good or not? We also need to have exploration, but at least maybe the simple answer to the first aspect of the question is trust the benchmarks, trust the challenges, etc. So that for today, what state of the art at least. I'm not sure I fully understood the second part of the question. Actually, I don't know if you want to.
The second part. What are the biggest bottlenecks to make sure that researchers are able to adopt these models? So, say you have a specific model that is meant for a specific use case. Not every single researcher that could be using that model is using it. How do you tackle that?
Yeah, I'm not sure. So bottlenecks. So maybe one part of the answer is that a real foundation model, at least what the field is trying to do, is meant to be a general purpose tool, meaning as soon as it's specific or good for one task, and we have many models, maybe AlphaFold, by the way, I mentioned it as a foundation model, maybe it's not the first version, but the more AlphaFold and similar models are being created, we realize that we can do good structure prediction by having a good foundation model of protein sequences. Okay, so I would say AlphaFold may not be a foundation model, but later generations are built on foundation models that can predict structure, that can predict function, that can predict interaction, etc. So I'm not sure it's a good answer, but at least I would say that in terms of foundation models, the ones I would trust or the ones I would investigate or try to build as a community are quite general purpose. Like, I can maybe mention the AI virtual cell initiative, which is really to say, can we have a foundation model, so AI system, that is just here to simulate cells, and we don't know for what purpose. But really, we just want it to be useful if you want to predict response to perturbation, or if you want to do cell type classification, or to do other things.
Thank you.
Cool. Yeah. Let's thank the speaker one more time.
Thank you.